I would like to go on with the cell, really. We need to have a very important understanding of the cell. We know that cells are renewed inside our bodies all the time. Our bone cells and eye cells and every other cell inside our body are being renewed. And what does this mean? It's built again. If we are to think of that as a city, really, and all the roads, the houses, and the infrastructure, and all the other details related with the city should be constructed again. And this results in codings, and those codes provide the right formula for construction of the right organelles, the right materials. So all are built over and over again. It's so extraordinary, really, because all these building blocks of a cell should be renewed and should be formed just from scratch, and they need to be established again. And we know that when a certain construction is being manufactured, is being built, actually, different role players just play their role. They just carry out their assigned tasks. And this is also true for the cell, because, for instance, while a manufacturing system is established inside the cell, all the corrections are made, all the controls are made. And for instance, when we look at the DNA molecule has this formula, has this coding, which provides all the details of this entire mechanism inside the cell. We know that this DNA formula is really very important, and it needs to be preserved without ever any harm given to it. So it's just placed in the cell's nucleus in a special place. And if there were any harm given to the DNA molecule, that would be fatal for the body. Because if there's any error with the protein molecule, that could be really corrected, or that would be entirely demolished and the new protein molecule could be formed. But if there's anything wrong with the DNA molecule, actually, that could not be corrected. And we know that if the DNA molecule is changed, then it turns out to be a cancerous cell. And if it keeps on reproducing, then that would mean cancer to reproduce throughout the body. This means that while the DNA molecule is transcribed, actually, we know that there are different stages of control. And we know that for the 100 trillions of cells, there should be no harm given. And we know that that harm should be prevented in such a way that at all stages there should be control and correction mechanisms, repair mechanisms. And the DNA molecule passes through different stages for correction. And I would like to talk about the fifth stage. We have a DNA molecule here, and the DNA consists of 4 nucleotides. And these nucleotides just make up the formula of the DNA molecule. In certain instances, what happens is the DNA may be subjected to radiation, and that radiation could be resulting in a kind of a harm on the DNA molecule. So what happens is thymine is not paired with the right nucleotide, but it just finds another nucleotide and makes a pair. And this is definitely a wrong sequence, and that would be resulting in changing of the code, the harming of the code, and DNA is not transcribed well, and then proteins are not formed, and that manufacturing system is actually devastated because of that harm. And what happens is there are the enzymes which just come into play when there is any such error in the building of the DNA molecule. And excision is the method that is carried out to prevent that error. For instance, excising means cutting. So enzymes that carry out this excision just come and carry out their work on the DNA molecule. They just come, and if there is anything wrong on the DNA molecule, they find the right place where that harm is placed on the DNA molecule. They excise it. They cut it off actually from the DNA molecule.
Şimdi bakın yukarıda gördüğünüz yerde hasar görmüş olan nükleotitleri görüyorsunuz. Hasar görmüş yeri değil sadece. İkinci alttaki şekilde koskoca bir alanı yuvarlak içine almış. Orayı koparıyor. Altta da gördüğünüz gibi o bölüm koparılıyor ve en sonunda gelerek tekrar yeni enzimler o koparılan o kadar geniş bölgeye tekrar nükleotitleri getirip yapıştırıyorlar. Bu çok özenli bir şekilde alınmış ekstra tedbirdir. Çünkü o gerçekten öyle. Oradaki nükleotit yandaki nükleotitlere zarar vermiş olabilir. O yüzden işte büyük geniş bir parça alınıp enzimler gelir bunları keserler. Oradan onu koparıp getiren başka enzimler vardır her zaman olduğu gibi. Yeni nükleotitleri getirip takan da yeni enzimler vardır. Böylelikle DNA yoluna devam eder işlemesine. Ama tamir mekanizmalarımız bunlarla bitmedi. Biz bu konuya inşallah devam edeceğiz. Her aşamasında Allah'ın Yüce Rabbimizin oradaki rahmeti, oradaki bize olan yani sevgisini hissetmek mümkün. Bakın her hücrede Rabbimiz bunu bize gösterir.